Welcome to the third lecture of the lecture series on the enzymes used in recombinant DNA technology. Today we are going to learn about the DNA ligases. DNA ligases are the enzymes which are going to seal the single stranded links between the adjacent nucleotides in a duplex DNA chain. So here the word which is important, it is what? It is adjacent nucleotide. And when you are saying that ligases, it is going to seal the next means what? It is going to synthesize the bond, and that bond, it is a phosphodiester bond. Now you can see, you can see here, this it is a double-stranded DNA, and in this double-stranded DNA, you can see there is a nick. Okay, so this nick, it is going to be sealed by the DNA ligases. Here at 5 prime end, here there is a phosphate, and at the 3 prime end, there is OH. Means what? Only it can seal the nicks, okay, which is having the 5 prime phosphate and the 3 prime OH. If it is going to be an inverse one, at that time it is not going to seal the nick. Now here in the second one, okay, in the second one, what you can see that there are the nicks in the both of the strands of the DNA. Such a nicks, they are also going to be sealed by the DNA ligases. And such a type of structure, it is going to be observed when you are going to sealing, okay, when you are going to seal the cohesive ends. Okay, so the cohesive ends which are going to be generated by the enzymes like the E.coli and T4 okay, that enzymes, they are generating the overhang sticky ends or the cohesive ends, and that cohesive ends, it can be sealed with the help of the DNA ligases. But the condition, it is what? Both of the nucleotides, they should be placed adjacent to each other. If they are far away from each other, then the nick, it is not going to be sealed. Okay. Now the next one, there are the types of DNA ligases. Okay, in the recombinant DNA technology, most of the time there is a use of the DNA ligases, and that DNA ligases, they are the E. coli DNA ligases and the T4 DNA ligases. E. coli DNA ligases which are isolated from the E. coli cells, and the T4 DNA ligases are the DNA ligases isolated from the E. coli cells infected with the T4 bacteriophage. Now here coming towards this chart, okay, here there is the list of the DNA ligases which are going to be used, and the substrate of that DNA ligases, cofactors which are required, and the temperature at which they are going to show the reaction. So the first one, it is a E. coli DNA ligases. It can seal the cohesive termini effectively, okay, at the temperature, and that temperature, it is of about the 10 to 15 degree Celsius. Okay, as it is a E. coli, you may say that the sealing temperature, it should be about the 37 degrees Celsius, but it is not going to happen because these cohesive ends, they may not be stable at a 37 degrees Celsius. To have the stable hydrogen bonding between the cohesive ends, the temperature, it is going to maintain at a 10 to 15 degree Celsius. Okay, so that there is a placement of the nucleotides adjacent to each other. That is a 5 prime and 5 prime phosphate and the 3 prime OH. Now they are adjacent to each other, and the nick, it is going to be sealed. These E. coli DNA ligases, okay, they can only seal the blunt ends only and only when there is an addition of the condensing molecule. Without condensing molecule, E. coli DNA ligases, they cannot do the blunt end ligation. And these DNA ligases, they are also not going to do the ligation into the DNA RNA hybrid. They cannot also seal the RNA nicks, okay, the nicks in between the RNA and RNA. These enzymes, they are requiring the NAD as an cofactor, okay, and at 37 degrees Celsius they can close the nicks. Okay, next one, here it is a T4 ligases, okay, T4 DNA ligases. This T4 DNA ligases, they can seal the cohesive termini, they can seal the blunt ends, okay, they can also do the ligation in between the DNA and RNA. They can also do the ligation between the RNA molecule, and here they are requiring the ATP as an cofactor. And T4 DNA ligases, they are going to seal the nicks into, or they are going to seal the cohesive termini at the temperature, and that temperature, it is a 4 degrees Celsius. It is going to close the nicks at the 37 degrees Celsius, and blunt end ligation at the 15 to 25 degree Celsius. Next enzyme, it is a ligases from the thermophilic bacteria, and these ligases from the thermophilic bacteria, it is only going to do the cohesive and terminal ligation. They are unable to do ligation of the blunt ends, DNA RNA hybrid and RNA RNA molecule. Like the E. coli DNA ligases, they are requiring the cofactor, and that cofactor, it is a NAD. And as they are the thermophilic one, okay, they are going to do the ligation at a higher temperatures. The cohesive end ligation, it is going to be done at 65 to 72 degrees Celsius, and it is going to do the closing of the nicks at the 37 degree Celsius. Now going towards the applications. Okay, so here the joining DNA molecules with the compatible cohesive end termini. This, it is the most important application of the DNA ligases in the recombinant DNA technology. Whatever the ends they are generated, the fragments which are generated, okay, that is your gene of interest or the foreign gene that you have to clone into the vector molecule, that has to be joined. Okay, for that joining of the DNA molecule, okay, you are requiring the DNA ligases. You can use the T4 DNA ligases as well as you can use the E.coli DNA ligases. Okay, but when there is, okay, when there are the blunt ends, okay, that blunt ends now they are going to be joined with the help of the T4 DNA ligases. Okay, the preferred enzyme it is a T4 DNA ligases, but with the help of the condensing molecule, the E. coli DNA ligases, they are also a choice of the enzyme. Now here there is, okay, for the joining or for the sealing of the synthetic linkers to the DNA molecules, there is a use of the DNA ligases. And thermostable DNA ligases, they are used into the a type of NPCR called as an ligase amplification reaction. Okay, so these are the number of applications of the ligases. I think you may have cleared the idea about the ligases. If you have liked my video, please subscribe my channel and also share my video with your friends so the knowledge it is going to spread. Thank you.